rolling circle DNA replication is a minimalist way to replicate DNA. If you're studying plasmid replication and bacteriophage life cycle, you should understand this concept. And if you're a researcher, this concept is applied in a technique called rolling circle amplification. So it also has practical utility. Since this was discovered in the context of bacteria, I should point out that it is different from the genome replication in prokaryotes, which uses theta mode of replication, where the template is circular DNA. In contrast, the linear chromosomes of eukaryotes have a multi-replicon style of replication, and telomeres have it slightly differently. I have separate videos explaining all of them. The theta mode, used to copy bacterial chromosome, starts with a single initiation point called origin of replication. In E. coli, we call it the ORI-C. Then a bunch of initiating proteins assemble at the origin and expose the single strands of the DNA, which is called the replication bubble. These single strands then become targets for replication fork assembly. The bubble expands as the replication fork moves. And this looks like the Greek letter theta. So we call it the theta mode of DNA replication. The Greek letter version of rolling circle is sigma mode. The symbol has a circle that looks like it is rolling on a surface. To understand rolling circle mode of replication, I will use M13 and PhiX174 bacteriophage DNA as the example. Bacteriophages are viruses that infect bacteria, and M13 and X174 are classified as coli-phages, because they infect a bacteria called E. coli. Let's start with the M13 phage, which looks like a submarine. The process starts when the virus injects its DNA into the bacteria, and then the virus DNA has to replicate. The DNA is circular and single-stranded. This single-strand DNA that is injected is called the sense or plus strand. Although most of the DNA is single-stranded, a small portion of it forms a hairpin structure, because of a 57 nucleotide palindromic sequence. This is the single-strand origin of the viral DNA. This hairpin is the only double-strand feature in this DNA. And as soon as the M13 injects the DNA into the bacteria, the single-strand binding proteins all cover up the DNA. This offers protection to the DNA against degradation. The fun part starts when the SSO and the SSBs somehow recruit RNA polymerase to this hairpin structure. The polymerase makes RNA, which disrupts the hairpin structure, and you end up with a DNA-RNA hybrid region. The RNA is only 25 nucleotides in length because the RNA polymerase is unstable on the single-strand templates, and it terminates transcription. We saw this in the mitochondrial DNA replication as well. The RNA forms the primer for the DNA polymerase III, which can extend it into DNA and copy the plus strand template. This also removes all the single-strand binding proteins from the template. When it comes full circle, the DNA polymerase III terminates, and polymerase I, which has the kleno fragment in the Fen domain, joins instead to remove the RNA primer. Then ligase joins and stitches the ends together. This results in a complete minus strand. We start with a single-strand DNA, and now we have a perfectly double-stranded DNA. This form of viral DNA is also called the replicative form 1, which is characteristic of a duplex DNA that can supercoil and do all sorts of molecular gymnastics. You cannot really supercoil single-stranded DNA, well, because it is single-stranded, and the fact that single-strand binding proteins will not allow it. Okay, now because this is a double-stranded DNA, the viral genome itself becomes activated. And since most of the genes are transcribed from the minus strand, the replication of plus strand results in transcription of most all genes in the genome. We will only talk about 3 genes, the gene 2, 3, and 10, which makes proteins 2, 3, and 10. Yep, they're labeled as simple numbers. It all starts with the gene or protein 2, which cuts, or should I say, nicks the plus strand. I will refer to the positions in context of the SSO, which is now double-stranded. The nick happens not too far from the SSO, and this position is the double-strand origin. The protein remains bound to the 5' end of the nicked plus strand. This form of duplex is also called replicative form 2. The older form was characteristic of supercoiling. This one is characteristic of the nick, which prevents the duplex from supercoiling. This means that GP2 is a site-specific endonuclease, but it also has links with the 5' phosphate now, so it is stuck. GP2 also has other functions. It will recruit helicase and DNA polymerase III at this nick site, along with the help of GP10 protein. So here's a representation which eventually leads to the helicase moving along and unwinding the DNA, while the DNA polymerase uses the 3' end, which is free, on the nicked plus strand to make new DNA. So essentially what I mean to say is that when the helicase moves, the 5' end in the front, and thereby the entire plus strand, is unzipped. But at the same time, a new plus strand is immediately recovered by the DNA polymerase. And now if you imagine that this process continues, which means that the helicase and polymerase make a full circle of the DNA, one full copy of plus strand will be unzipped. Since this is a single-stranded DNA, the SSO will again recover a hairpin structure, while the rest of it is protected by the SSBs. At this SSO, other proteins like GP3, Rec family proteins, and a bunch of other proteins can help release this one copy of plus strand, or even terminate the replication on the minus strand. If it decides to terminate, then the same old machinery can fix the final DNA ends. After termination, you get this double-stranded DNA molecule with the plus and minus strands. And this would be in a supercoiled form. The other product is the plus strand, covered in SSBs, and has its own signature hairpin. But why stop here? The helicase and polymerase could actually keep making more and unwinding the plus strand. So depending on the number of repeat cycles, you get many copies of the plus strand. Actually, this RF1 can be converted into RF2 if the GP2 protein is still around, and this might start another round of replication. And likewise, the plus strand can again recruit RNA polymerase and start yet another round of replication cycle from the beginning and make more RF1 and then more RF2, and the cycle continues. Since this is a viral DNA replication, the plus strand is also packaged to produce more M13 phage particles. So this is a typical rolling circle, where you have a double-stranded DNA being unzipped and rebuilt at the same time. As you saw, this occurs continuously. There is no lagging strand or Okazaki fragment structure in this replication, at least in the case of M13 phage. The PhiX174, however, is not the same. Instead of a submarine, it also looks different. At the DNA level, it is still single-stranded DNA and circular just like the M13. But there's a twist in the rolling circle mechanism. It uses a variation called loop rolling circle mode, and it uses specific proteins called primosomes to achieve that. Let's dive deeper. It seems to start out like the M13 example. There's an SSO, which is a 70-nucleotide sequence in this case, that forms a hairpin. The SSO has a name. It is called the PAS site, which stands for Primosome Assembly Site. So it may look like the M13 example, but it is not. The PAS site recruits a pre-A protein, which is a translocase on the single-strand DNA. And then two other accessory primosomal proteins join the pre-A. This establishes the recognition of the SSO. To this SSO, the DNA B and DNA C complex, which is the helicase and its loader protein, join. This occurs with another loader protein called DNA T. And all these proteins bind to the pre-A, B, and C to form the pre-primosome complex. The equilibrium then shifts forward when the primase also comes in and binds to the helicase. This converts this structure to a functional primosome complex. After loading the helicase, the loader proteins are released from this complex. So the primosome is a complex of three primosomal proteins, the helicase and the primase. You can think of this entire structure as a giant translocase. And when it translocates, the primase starts adding primers at random location as the complex moves on the single strand. Just a side note, you don't need the primase for translocation, so both pre- and full primosome complexes are capable of movement. And as I said, during the translocation of primosome, the primase will add RNA primers throughout the DNA. Note that the helicase moves in a 5' to 3' direction, but the primase synthesizes the primer in the opposite direction. I have a full video linked in description that addresses this contradiction. Once these primers have been laid down, the DNA polymerase III will pick up at the 3' ends and start synthesizing the minus strand. And when it is done, the same old enzymes will make sure to remove the RNA primers and ligate the disjointed ends. This will result in a fully double-stranded DNA molecule with a plus and a minus strand. This is also the replicative form 1, which can undergo supercoiling. Now, the important stuff to note is that the primer making and DNA replication is temporal, meaning that some regions are replicated first, depending on when the primer was laid down. This is important because next to SSO, where the first primer is laid down, the first gene is present. Gene A is the first to be converted into a double-stranded DNA. And close to the promoter of the gene A is the double-strand origin. All this means is that when gene A is in a double-stranded form, it can be transcribed immediately and eventually form a protein. This gene A protein binds to the DSO, which contains its recognition sequence. Note that binding occurs at the minus strand, so you cannot have this scenario with the plus strand alone. After binding to the minus strand, the protein nicks the plus strand at the DSO. On top of this, the protein A has affinity for the primosome complex as well. So after laying down primers and translocating, the primosome does not just disassemble. The protein A is an endonuclease. After nicking, a free 3' and a 5' end is generated, except that the 5' end is covalently bound to the protein A. There are a couple of tyrosine in the core of the protein A that mediate this sort of linkage. And the primosome is still present around the nicked site. Then, the protein A recruits a helicase called Rep helicase onto this primosome. Pre-C also has some affinity to this Rep helicase. Rep helicase is a bacterial helicase for special occasions like replication restarts. And it is monomeric, in contrast to the DNA B helicase, which is a hexamer. Now, since there is a nick in this duplex DNA, this is a replicative form 2. Now you have protein A, Rep helicase, primosomal proteins, DNA B helicase, and primase, which may actually dissociate away. It is not that important anymore. In addition, the available 3' end means that a new DNA polymerase can be loaded here to start the DNA synthesis. You have to realize that all these proteins are dynamic, and especially that the primosome and protein A are physically a single complex that moves together. And the DNA polymerase is perhaps also associated with this, behind the complex, making new DNA. Now, because the 5' end of the nicked plus strand is linked to the protein A, and protein A is linked to the primosome, when the helicase unbinds the plus strand is looped out. The looped out DNA is protected by the single-strand binding proteins. So new plus strand DNA is being made as the old DNA is unzipped and looped out by the helicase. When this moving complex comes full circle, or at least once it has passed the DSO, a new protein A can bind there and make a cut at the plus strand. This means that when the primosome returns to the starting position, the plus strand is released from the loop because of the nick. And the new protein A that made this cut can start the same process of replication once more. And if it were to terminate, you end up with a double-stranded DNA, which may be in an RF1 form, which can transition into RF2 form, depending on the availability of the protein A. And the other product is the looped-out plus strand, which is joined at the cut site. The mechanism for joining the ends involves protein A, where these two tyrosines do the magic, allowing for a nucleophilic attack from the hydroxyl on the 3' end to the phosphate on the 5' end. And this eventually results in a circular plus strand. Depending on how many cycles you go through on the RF2, you can have many copies of this plus strand, which can either restart the primosome assembly at the pass site, or it can be packaged to make new virus particles. We discussed that the M13 rolling circle was fully continuous on both plus and minus strands. In contrast, the PhiX174 uses discontinuous replication for the minus strand, which is sort of reminiscent of lagging strand DNA synthesis. And the plus strand here is generated by a continuous leading strand DNA synthesis style. So even within rolling circle modes, you can have very different ways to perform DNA replication. And that's all for this discussion. I hope it was useful, and as always, I will see you in the next video.